So hi everyone, this is Harshita. So today we are starting with the series of recombinant DNA technology. So it is also known as genetic engineering. So the basic definition of RDT is it is a set of techniques that enables DNA from different sources. The sources could be yeast cells or bacterial cells that has to be identified, isolated, and recombined. So while these steps are performed so that we can introduce a new characteristics into the host cells. The very first recombinant DNA molecule was developed by the scientist Wahlberg. That was taken from SV40 virus and lambda base. The basic characteristic of a recombinant DNA technology is DNA cloning. So you all must be aware with the definition of DNA cloning that it is a method or production of identical DNA molecule from a single ancestral DNA molecule. The essential characteristic of DNA molecule is it should be selectively amplified. Amplification means it results in the large number of copies of a DNA molecule. So basically there are two different types of approaches. With the help of them we can done a DNA cloning. The first one is cell based and the second one is cell free. So we will dealing both of them one by one. The basic steps of cell based DNA cloning is that attaching a foreign DNA molecule or DNA fragment to a DNA sequence. The DNA sequence that has to be attached with them should be capable of independent replication. Then the recombinant DNA is transferred to a host cells. So recombinant are the DNA molecule that has autonomously replicated DNA and they have inserted element that is known as hybrid. So these both are done with the help of covalent ligation that is done between the targeted DNA and the replicon. Replicon could be any sequence that is incapable of independent DNA replication. So the next step is transformation. The recombinant DNA molecules that are produced has to be transferred into the host cells. So this process is known as transformation. So we will be seeing an example of plasmid vector where we will doing a transformation with help of plasmid. So here comes our cloning vector. I'm writing it in a short form, that is CV, and other one is our target DNA. So with the help of restriction enzyme, we cut cloning vector, and with the help of ligase enzyme, we will be ligating both of them. So here comes our recombinant vector. So other thing is we have a non-recombinant vector. So we will be dealing with the three case. We are taking bacterial cells in which transformed cells are taken. This is a chromosome of bacteria. So this is a recombinant vector and this is a non-recombinant vector, and in the third case there is nothing. So in the next steps we will be taking a cell with the transformed cells. So after that these two cells are taken, then they are again screened and only cells with the recombinant vectors are taken. So the recombinant cells, then it is amplified so that we can produce a large number of copies. Then another step come here is selective propagation of a cell clones. So it is done basically in two stages, in which the transformed cells are spread out on a agar medium so that we can encourage the growth of well separated colonies, and then colonies are picked from the plate and these can be further expanded in a liquid culture. And the last step is isolation of recombinant molecule, or we can say that DNA clones. Now, another step of, or the method of DNA cloning is cell-free approach. So basically this is done with the help of PCR, polymerase chain reaction. The difference between both of them is in the cell-free process, it is entirely an in vitro process. So what is PCR? You all are aware that the short primers are taken and these are amplified. So it is a very rapid, inexpensive and simplest method of copying sequence specific DNA sequence. So our next topic is enzymes for DNA manipulation. So basically there are basically four types of DNA enzymes that are basically used. That is template dependent DNA polymerase. So if you have gone through cell biology, you must be known with the basic meaning of DNA polymerase, that it is a polynucleotide that catalyzes the replication process. Second is nucleases enzyme. Third is end modification enzymes. And the last one is ligases. So we will be dealing with all four of them one by one. The template dependent DNA polymerase, it synthesizes new polynucleotide that is complementary to an existing DNA template. So there are three types of DNA template dependent DNA polymerase, that is DNA polymerase 1. So it has all the three activities, that is 3' to 5' and 5' to 3' exonuclease activity. And it also have 5' to 3' polymerase activity. The second one is reverse transcriptase. So it is a RNA directed DNA polymerase that synthesizes DNA from RNA. Third one is tag DNA polymerase. So it is usually operated at 72 degrees Celsius and it is derived from a thermostable bacterium, that is Thermus aquaticus. So it has both 5' to 3' polymerase activity and 5' to 3' exonuclease activities, but it lacks 3' to 5' exonuclease activity. So our second enzyme, nucleases. So it is an enzyme that degrades nucleic acid by breaking the phosphodiester bond between them that link one nucleotide to the another. So basically there is a exonuclease enzyme and a endonuclease. This is a type of nucleases enzyme. Nucleases could be RNAs H that degrade RNA, DNAs that degrade DNA. So it can also degrade single-stranded DNA. It can also degrade or break a double-stranded DNA. So exonuclease, it removes the nucleotide at the end of a nucleic acid. At the end of a nucleic acid. And in case of endonuclease, it breaks internal phosphodiester bond between the nucleic acid. So there are basically there are any particular exonuclease that usually attack either the 5' end or the 3' end, but here comes our exception, that is BAL31. It attack both 5' end and 3' end of the nucleic acid. So we have a types of nucleases, that is mung bean nucleases, but it degrades single stranded DNA and RNA. It will digest single stranded nucleic acid, but it will leave intact any region which is double stranded in the case of nucleic acid. Then here comes our S1 nucleases. It degrades RNA and a single stranded DNA, but it doesn't degrade double stranded DNA and or RNA DNA hybrid. So it will also cleave an opposite strand or nick of the complement. So third one is RNase-H. You all must be aware that it is a endonuclease which digests single stranded RNA at a 3' end. So it could, at the 3' end there could be a pyrimidine residue. You all are, I must say that you all must be aware of the pyrimidine and purine. The in under pyrimidine, here comes our cytosine, thymine, and uracil. So that is all.